Welcome back, dear students. We have already started with chapter number 2. It is sexual reproduction in flowering plant, that is angiosperm. In the last session, we have discussed a sexual reproductive part of an angiosperm, and that is a flower. We have understood the different parts of a flower. Flower contains main four valves: calyx, corolla, androecium, gynoecium. From these four valves, calyx and corolla, they are accessory and the sterile valves. They do not involve in sexual reproduction directly, whereas androecium and gynoecium, they are fertile valves and they involve in a sexual reproduction. Androecium is a male reproductive valve, its individual unit is stamen, and the carpel is a female reproductive valve, its individual, and the gynoecium is female reproductive valve and its individual unit is carpel or pistil. Okay? See, any living organism who reproduces sexually, in that living organism, whatever the type of organs they have, but the basic events in sexual reproduction involves, that remains the same. Means it includes prefertilization, fertilization and post-fertilization. Today we will start with prefertilization and we will just finish, or we will learn a structure of male part of a flower, and that is androecium. And androeciums are made up of a stamen. Okay? We will learn how male gametes are going to be produced. Okay, and then in the next session we will learn how the female gametes are going to be produced. Okay, and that we will finish with gametogenesis. But to understand how the male gametes are going to be produced, we should also know the structure of male reproductive part in detail. So now students, you know that which is the male reproductive valve? The male reproductive valve is androecium. And this is a valve, a whole layer. Okay. And the individual unit of this androecium, it's what we call as stamen. Okay. Now this stamen, what is the another name of stamen? What we have seen in the last session, it is also called as microsporophyll. Okay. What's another name of stamen? It's also called as microsporophyll. Let's see the structure of stamen, how the stamen look like. You know that stamen mainly consists of two parts. First is called as filament, and the second part of a stamen is called as an anther. So see, if I draw a structure of a stamen, it contains a slender stalk-like portion. Slender stalk-like portion. This portion is called as filament. And at the terminal end of it, it contains anther. Usually, this anther, it is bilobed. Okay, students. So this is two parts of a stamen, filament and the anther. Filament is a slender part of a stamen, and at the terminal end of a filament there is a presence of bilobed structure, what we call it as anther. Filament is a sterile part of a stamen and the anther is a fertile part of a stamen. This anther and filament, they are connected by connective. See, now you see the different types of a flower. The length of this stamen may going to vary from flower to a flower, or it may vary from plant species to a plant species. Okay. Here I have shown just a one stamen, but it is not always necessary that the plant or the flower contains only a one stamen in a flower. Okay. It may happen that the number of stamens which is present in the flower, they may also vary. So ultimately, the number of stamen in a flower and the length of a stamen in a flower may vary from a plant species to a plant species. Okay. So see, this is a filament, it is a sterile part, and this is a anther, it is a fertile part. The male gametes are produced over here, or the pollen grains are produced within it. So let's see how the pollen grains are going to be produced. Okay. So now we will start. But to understand how the pollen grains are going to be produced, we should know the structure of this anther, a fertile part. Now, if I took a transverse section of this anther, then how the anther looks like? If I took a transverse section, it's look like this. This is a part of filament. And when you see it from the top in the transverse section, you will see such kind of a structure. Okay, so these are the structures which are present of an anther that can be seen in the transverse sections. If you see this transverse section of an anther, there is a presence of a longitudinal groove which is running the lengthwise. This is the longitudinal groove which is running the lengthwise, and such kind of a structure you can observe. Okay, now I just enlarge this portion of an anther, and then we will discuss the internal structure of an anther. Okay, so I just enlarge this much part. Okay, how it look into this. So if you see the anther, it consists of, it's a bilobed. Means it contains how many lobes? 2 lobes are present. Each lobe contains 2 theca. How many theca are present? 2 theca are present. So it is also called as dithecus. Okay, I again repeat how the anther looks like. Anther is bilobed, means it contains 2 lobes. Each lobe contains how many theca? 2 theca are present. So it is called as dithecus. Okay, so what is the characteristic you have to remember of an anther structure? It is a dithecus structure. Now if you see its sides which are present, that how many sides are present? There are 4 sides. 1, 2, 3 and 4. Means the anther is a tetragonal structure because it has 4 corners or 4 sides are there. Barabar? So that's why it is called as a tetragonal structure. Each side it contains microsporangia. This is a circular in nature. So this is a microsporangia. Okay. So this is about internal structure of an anther. I again repeat, in the internal structure of an anther. When you see the internal structure of an anther, or when you learn the internal structure of an anther, the anther contains how many lobes? There are 2 lobes are present, so it's called as a bilobed. Each lobe contains how many theca? 2 theca. So that's why it is called as a dithecus structure. Okay. How many sides or corners are present? 1, 2, 3, 4. It's 4 sides and corners are present. So that's why it is called as a tetragonal structure. Okay. Each corner contains microsporangia. Okay. Microsporangium. This is a singular. Here it's a 4, so it's called as microsporangia. Okay. Now we will learn the individual structure of this microsporangia. How this microsporangia looks alike, or what are the structural components of a microsporangium. So I just enlarge this much part. What it contains? Microsporangium. This microsporangium, it contains 4 layers. Okay, which are the 4 layers? The first layer of a microsporangium, it's called as epidermis. The second layer of microsporangium, it's called as endothecium. The third layer is called as middle layer, and the fourth layer is called as tapetum. So how many layers of a microsporangium? The microsporangium has 4 layers: epidermis, endothecium, middle layer and the tapetum. These are the 4 layers which are present into a microsporangium. If you see the shape of this microsporangium, it is circular in outline. It is a circular in outline. Specifically, when you observe in a transverse section, it looks like a circle, so it's a circular in outline, and it contains these 4 layers: epidermis, endothecium, middle layer and the tapetum. Okay. So now I just draw the structure, this much part. I am just drawing this much part. Okay. This is the first layer, second layer, third layer and the fourth layer. So this is the first layer of it. Okay. The first layer of it, it's called as epidermis. Then there is a second layer, then there is a third layer, and then there is a fourth layer. The second layer is called as endothecium. The third one is called as middle layer, and the fourth one is called as tapetum. Each one is made up of different types of cells. See, these are the different types of cells which are present in it. Okay. Now from this four layer, the function of this first three layer, epidermis, endothecium, and the middle layer, they are two functions. First, the function of this three layer is protection, and the second function, that is, they help in dehiscence of pollen grain. Means in simple term you can say that they help in a release of pollen grain. So what is the function of first three layer? Epidermis, endothecium, and the middle layer. Their function is the protection of developing pollen grain, and the second one is they are helpful in the dehiscence of a pollen grain, means a release of pollen grain. The fourth layer, it's called as tapetum. The function is it provides nourishment or nutrition to an offspring. This is the fourth layer. It provides a nourishment or nutrition to an offspring which is present inside. In the center, we will see how this pollen grain will be developed. We have just seen the four layers. But in the center, there is a formation of a pollen grain occurs. How the formation of pollen grain occurs, that we will see. But to develop this pollen grain, it needs a nutrition. That nutrition is provided by the fourth layer. It's called as a tapetum. Okay? They provide the nourishment to a developing offspring. Now, if you see the cells of this tapetum cells has a dense cytoplasm, and it has more than one nucleus. Usually it contains a two nucleus. One cell contains a two nucleus. That is in the case of tapetum. So what are the characteristics of the cells of a tapetum? The cells of tapetum has a presence of dense cytoplasm, and it has more than one nucleus. Usually two nucleus is found. So you can say that the tapetum cells are binucleate. Means it contains a two nucleus. Okay. So these are the characteristics of a microsporangium. Okay. I again repeat how the anther looks like, or what is the internal structure of an anther. As usually you know that anther contains how many lobes? Two lobes are present, so what we call it as bilobed. Each lobe contains how many theca? This is a one lobe, it contains two theca. This is the another lobe which contains two theca. So it's a two theca, each lobe contains two theca, so it's called as dithecous. If you see the sides and corners, so how many sides? Four sides, so it's called as tetragonal. In each corner there is a presence of microsporangium. Okay, microsporangium, how it's look like? Circular. It contains how many layers? Four layers: epidermis, endothecium, middle layer, tapetum. Okay, the function of first three layer, epidermis, endothecium, and the middle layer, is the protection, and it helps in dehiscence of anther, means release of pollen grain. Pollen grain. Fourth one is tapetum. Its function is it provides a nourishment or nutrition to a developing pollen grains. The cells of tapetum are such that it has a dense cytoplasm and it has more than one nucleus. Usually two nucleus are present. Clear? Now, in the center of this microsporangium. So this is the center of microsporangium. They have a homogeneous cells. And these homogeneous cells, they are compactly arranged. How they are arranged? Compactly arranged. So the center of microsporangium, this is the center of, I show over here. This is the center of microsporangium. It contains compactly arranged homogeneous cells. Okay, compactly arranged homogeneous cells which are present over here, and they are called as, or like over here, they are called as sporogenous cells. What they are called as? Sporogenous cells. These sporogenous cells can be found when the anther is young one, when it is developing, when it is growing. At that time, the center of microsporangium, you can observe compactly arranged homogeneous cells. What is the name of these cells? They are called as homogeneous cells. And remember, when it is observed, they are observed when it is young or it is growing. Clear? So that is all about the characteristic of internal structure of anther. Still the pollen grain is not developed. We have just seen the structure. Now from this structure we have to learn that how the pollen grain is going to form. Okay? So we will start with a process how the pollen grain is formed. Clear about the structure? If you are clear about the structure, then and then you can understand how the process of formation of pollen grain occurs. Okay? So let's start how the process occurs. So to understand how the pollen grain occurs, we have to learn one process, and this process is called as Microsporogenesis. What we call it as Microsporogenesis. Now you know that this is a word which is made up of two terms, microspore, and the second one is genesis. I hope all of you know the meaning of genesis. That's formation. So the process of formation of microspore, okay, this is called as microsporogenesis. So what is the definition of microsporogenesis? It's a process of formation of microspore from pollen mother cell, short form is PMC, pollen mother cell, through by which process? Through meiosis. Okay, that is called as microsporogenesis. I again repeat, what is the definition of microsporogenesis? It is a process of formation of microspore from which cell? Pollen mother cell. Through which process? Through meiosis. It is called as microsporogenesis process. This microspore can also be called as pollen grain. What's another name of microspore? Pollen grain. So we have to learn that how this pollen grain are going to be formed. Okay. So you know that we have learned the structure of microsporangium. Okay. This is a circular in outline, and in the center of this microsporangium there is a presence of sporogenous tissues, or there is a presence of pollen mother cell. This is called as pollen mother cell which is present in the center of microsporangium. What's called as pollen mother cell. This is a pollen mother cell. Pollen mother cell, it contains how many number of chromosomes? It contains a 2n number of chromosomes. Okay, because it is a normal diploid cell. It is a diploid cell. Pollen mother cell. Remember what is a ploidy? Ploidy. You have to remember ploidy because it's important. It is asked in some competitive examination. Ploidy. Okay, whether it's a diploid or haploid, this is called as ploidy. So it contains how many number of chromosomes? The pollen mother cell. Pollen mother cell, it contains a diploid number of chromosomes. Now what happens? This pollen mother cell, it divides by a process called as meiosis. Meiosis, you know that it is a reductional cell division, and by this type of a division, how many daughter cells are produced? 4 daughter cells are produced. Okay, so here it divides and 4 daughter cells are produced. 1, 2, 3, 4. Okay, these 4 daughter cells are produced. Here it is a meiosis. So the number of chromosome in this daughter cell gets half. Here 2n, so it gets half. N. Okay, but see, this 4 cell remains attached. It form a cluster, a group of 4 cells. They are not separated from each other. So this group of 4 cell or the cluster of 4 cell which are remains attached, this is called as microspore tetrad. What we call it as microspore tetrad. Okay, so this is the first step in microsporogenesis. What is the first step? Here in microsporogenesis, pollen mother cell, it contains how many number of chromosome? 2n number of chromosome. It divides by which process? Meiosis. It's a meiosis, so obviously how many daughter cells are produced? 4, containing half number of chromosome than the parent. So parent has 12 number of chromosome. This daughter cell has 4 number of chromosome. This cell doesn't remain separated. They remains attached together initially. Okay, it forms a cluster of 4 cells. This cluster of 4 cells, it's called as tetrad, microspore tetrad. Okay, now this microspore tetrad, it mature and it undergo a dehydration process. Okay, they become so mature. Okay, once they become mature, all these 4 cells becomes separated. And the 4 cells which are separated, they are considered as microspore. Okay. So after maturation and dehydration, this 4 cell gets separated and the microspore formation occurs. And this process is called as microsporogenesis. This microspore, what we can also call it as pollen grain. And thus, pollen grain are formed. Okay. So the process of formation of microspore from pollen mother cell by a process called as meiosis. I again repeat how the process occurs. In microsporogenesis, the pollen mother cell, which contains 12 number of chromosome, divides by mitosis and form the 4 cells. Each contains n number of chromosome. Remember, that means they are haploid. But initially they remain attached together, form a cluster called as microspore tetrad. Why tetrad? Because it's a cluster of 4 cells. Okay. Now once it becomes a mature, it gets dehydrated. There is a loss of water, dehydration. And so that's why these microspores are separated. And thus there is a formation of microspore or pollen grains are formed. So from one sporogenous cells, pollen mother cell, 4 microspores are produced. But you know that in the center of microsporangium, there are lots of sporogenous cells. If one cell of sporogenous cell or pollen mother cell generates a 4 pollen grain, then many cells generates lots of pollen grain and thus many pollen grains are going to be produced. And you know the reason why there is a need to generate the many pollen grain, because this pollen grain has to be transferred to a stigma or the carpel, and during the transfer, gamete transfer, okay, many gametes gets lost, and to compensate the loss, the living organism has to generate the male gametes in a higher number, and so that's why the pollen grains are produced in a higher number. Okay. So what we have done until now, we have finished with internal structure of an anther, or we have seen the structure of anther, basic structure of anther or internal structure of anther. We have also learned what is the structure of microsporangium. Okay, and we have seen the third process, that is called as microsporogenesis, means how microspore or the pollen grain is produced. Okay, now we have to learn the structure of this pollen grain and how the male gametes are going to be produced within this pollen grain, that we have to learn. So let's start about the characteristic of pollen grain, or you can also call it as microspore. Okay, what are the characteristics and how it's look like? If you see this pollen grain usually it's a spherical in shape, and the diameter of this spherical pollen grain is 25 to 50 micrometer, means it's a very small. Okay, this is the diameter of this spherical pollen grain. Usually it's going to be found. Okay, so what is the shape of this pollen grain? It's a spherical with a diameter of 25 to 50. But it is not always that the pollen grains are spherical only. We can also observe the different shapes of a pollen grain. And what are the different shapes? It may be a oblong sometimes, it may be also oval, or it may be also disc-like. Such kind of shape of pollen grain can also be found. Okay. Now, this spherical pollen grain, usually they are produced in a large numbers and look like the particles of dust. How it's look like? Particles of a dust. You will observe it into a practical, because you have to see this pollen grain under a microscope. So when you dust it out, it will just like the small particles of a dust. So it's a look like a dust particles. Now, if you see the pollen grain, they are uninucleate. Means it possesses how many nucleus? 1. and that nucleus contains how many number of chromosome? Because you already know that microspore contains how many number of chromosome? N. So obviously that pollen grain, what we also call it as microspore, contains how many number of chromosome? It's N. Means what is a ploidy of pollen grain? It is a haploid. Okay, what is a ploidy of pollen mother cell? It is a diploid. Okay, what is a ploidy of microspore? You can say that in other term, it's a haploid. So it is a uninucleate. It is a unicellular, means it is only just made up of a one cell and one nucleus. If you see its surface, the surface may be smooth or it may be spiny. Like it may be a smooth, it is spherical. Okay, or it may be a spiny. Like this. So this is the basic information regarding a pollen grain. Okay. So what's another name of pollen grain? Pollen grain can also be called as microspore. Usually it's a spherical in shape with a diameter of 25 to 50 micrometer. Sometimes it can also be observed in different shapes like oblong, oval, disc-like, etc. If you see it, it's morphologically look like the particles of a dust. It is unicellular, means just made up of one cell. One pollen grain is made up of one cell and contains how many nucleus? One nucleus. That's why it's called as uninucleate. Contains how many number of chromosome? N number of chromosome. If you see it's a surface, the surface may be either smooth or it may be a spiny. Okay. So this is what the pollen grain looks like. I just enlarged the structure of pollen grain. Usually it's a spherical. Okay, and contains an N number of chromosomes. This is a pollen grain. Are you agree that it is a plant cell? Yes, it is produced from a plant. It is produced from flower. It's a part of a flower. Okay, and flower is a part of a plant. So obviously this is a plant cell. And as you already know that the plant cell contains two layers surrounding to it. The outer layer of plant cell, it's called as cell wall, and the inner layer of plant cell, it's called as cell membrane. How many layers of plant cell? Two, cell wall and cell membrane. Okay, so similarly it's a pollen grain. So pollen grain contains also two layers. The outer layer of pollen grain, what we call it as cell wall, and the inner layer, that's it, what we call it as cell membrane. Now this cell wall, the cell wall of pollen grain, it consists of two layers. Remember, the cell wall of pollen grain again consists of two layers. Which are the two layers? The outer layer of the cell wall of pollen grain is called as exine, and the inner layer of pollen grain is called as intine. Okay. So the outer layer is exine and the inner layer, what we call it as intine. Okay. This outer layer, exine, is made up of a specialized chemical, what we call as sporopollenin. Okay. The exine is made up of what? The exine is made up of sporopollenin. The sporopollenin is an organic material that is highly resistant material. Even strong acid cannot damage it, strong alkali cannot damage it, even it cannot be damaged by the high temperature. It is the strong resistant material which is found in the exine, and this is the reason that's why we can find a pollen grain in the fossils, because the fossils of a pollen grain can be easily preserved because of layer called as sporopollenin. Clear? So the cell wall of pollen grain consists of how many layers? Two, exine and intine. Which is outer layer? Exine is the outer layer. It is made up of which material? Sporopollenin. This is the organic material which is a resistance to a strong acid, strong alkali, and higher temperature. No one can affect it, or there is no one in the world has been known till date which can damage this layer. Now the second layer, this is called as an intine. It is an inner layer. This inner layer, it is made up of, I write over here. The inner layer, it's made up of cellulose and pectin. Okay, so the inner layer is made up of cellulose and pectin. Okay, but if you see this outer layer of exine, there are certain places in the exine where sporopollenin is absent, like this place. this place where the sporopollenin is absent. Where the sporopollenin is absent in the exine, this place, it's called as germ pore. Okay, from this place we will learn later on a tube-like structure will develop like this. I will show over here, tube-like structure. This tube-like structure is called as pollen tube. Because this layer is so hard that it cannot be broken by even strong acid, strong base, so there is no chances that the cell will emerge it out. Okay, cell will come out from it. So that's why there is a space naturally from where the cells will come out, and this space where the sporopollenin is absent, this is called as germ pore. Okay, remember what is a germ pore? It's a location on the outer wall of cell wall, that's exine, where the sporopollenin is absent. What we call it as germ pore, from where the pollen tube arises. Okay, so that's all about the two layers of cell wall, exine and intine. Exine is made up of sporopollenin, intine is made up of cellulose and pectin. At some places there is an absence of sporopollenin. The places where sporopollenin is absent, this is called as germ pore. And from this is a place where the pollen tube arises. Now the another layer of pollen grain, this is called as cell membrane. It is like usual cell membrane. It's made up of lipids and proteins. Okay, it's made up of lipids and protein, and inside that what is present? Cytoplasm. So the cell membrane or plasma membrane, the another name of cell membrane is plasma membrane, you already know that. That is surrounded by cytoplasm, or surrounded to a cytoplasm, not by, it's to, surrounded to a cytoplasm. And you know that pollen grain contains a nucleus that contains how many number of chromosomes? That contains n number of chromosomes. Clear about the structure or the layers of pollen grain? Okay. So that's about the layers of pollen grain. Now we move ahead. Now you know that this is a pollen grain. It contains two layers. Okay? There are certain places where there is an absence of sporopollenin. It contains n number of chromosomes. Now what happens to it? Or how the male gametes are going to be produced, or in that. This pollen grain, now it goes under a division, and this is a mitotic division. But here, during the division, the type of cells which is going to be produced is like this. One cell that is going to be produced is the bigger one. This is the one cell which is going to be produced, and the other cell which is going to be produced is a smaller one. Okay, the bigger cell which is going to be produced, it's called as vegetative cell. What we call it as vegetative cell. And the smaller cell which is going to be produced, it's called as germinative cell. Okay, so that division occurs. Here the division occurs by which process? Mitosis. Mitosis means obviously the number of chromosomes remains the same as a parent cell. So that vegetative cell contains an n number of chromosomes. Similarly, the germinative cell also contains a n number of chromosomes. Okay. But what are the characteristics? One characteristic of vegetative cell and the germinative cell. The vegetative cell is big. It's bigger in size. Apart from that, it has a large food reserve. Means it contains a lots of food, and this food is utilized, or that is going to be supplied to a male gamete for its survival. And it has irregular shaped nucleus. Okay, so what are the characteristics of vegetative cell? It's a bigger, it contains a food reserve or large quantity of a food reserve, and it has an irregular shaped nucleus which is present in it. Now the second one is germinative cell. It's opposite, it's a smaller one. Okay, it has dense cytoplasm. This is a characteristic of, it's a pollen grain. Okay, or germinative cell. So here two types of the cells are produced. One is a vegetative cell and the second one is called as germinative cell. Okay. Now the pollen grain gets released from anther. Usually in 60% of the angiospermic plant or angiosperm plant, the pollen grain is released at this stage. Means it's a two cell stage. One is vegetative cell and the another one is germinative cell. This is a two cell stage. At that time the pollen grain gets released. Okay. But in 40% cases what happens? This germinative cell, it again divides. Germinative cell again divides and it forms two cells. These two cells are going to be produced. What we also call it as male gametes. What we call it as male gamete. And male gamete contains how many number of chromosomes? N number of chromosomes. Each contains n number of chromosomes. That's why we have said gametes are haploid. Same, it follows the rule. Male gamete is haploid. And this is a three cell condition. One and this two, three. From in this three cell, one is vegetative cell and the two cells are gamete cells, male gametes. So there is a three cell condition that happens in the 40% of angiospermic plant. So the pollen grain is released either at a two cell stage or three cell stage. Okay, in 60% angiosperms, the pollen grain is released at two cell stage, means it contains vegetative and germinative cell. But in remaining 40% cases, this germinative cell again divides and form two gametes, male gametes. And at that time the cell gets released, means cell gets released or pollen grain gets released at three cell stage. Okay, one vegetative cell and two germinative cell. Okay, so that's about a development of male gamete or the male gametophyte occurs. Okay, so that is development of male gamete. So what today we have seen? We have seen the internal structure of anther, we have seen the structure of stamen, we have seen the structure of microsporogenesis, we have seen process by which a microspore of the pollen grain is formed, and we have also discussed a few characteristics of pollen grain. Some characteristic of pollen grain remains, but that we will discuss in the next session. Till then, you have to keep learning, you have to learn whole this process, and then we will go ahead in the next session. Thank you for watching. Keep subscribe this channel. Thank you.